this video, we will be focusing on the life cycle of HIV and the antiretroviral therapeutic options, or ART, therapy options available to ideally minimize viral replication, maximize immunocompetency for as long as possible for the patient, and prevent the transition to acquired immunodeficient syndrome or an AIDS diagnosis. So to start off this video, we'll first start by talking about the structure of the HIV virus. So HIV is an RNA retrovirus that contains 2 duplicate copies of a single-stranded RNA, which encodes the HIV genome, which includes 9 genes and 15 proteins. And these RNA strands are located inside the viral capsid, along with several enzymes that are essential for its viral life cycle, including reverse transcriptase. We also have protease and integrase. And the capsid is surrounded by the matrix, which helps ensure the integrity of the viral capsid. This is then surrounded by a lipid bilayer, membrane that makes up the viral envelope. So the viral envelope is decorated with glycoprotein, or GP120, and glycoprotein, or GP41, which are subunits of the envelope protein complex. So now that we have visualized the viral makeup, let's go over the key steps in the HIV life cycle and how HIV infection and replication occurs. So the first step is for the HIV virus to bind the host immune cells, specifically our CD4 positive lymphocytes, like our T cells, for example, which I'm showing you here. So this virus will bind via its envelope protein complex, being our GP41 and our GP120, which will interact with our CD4 receptors, which is the primary interaction, and this is followed by its secondary interaction with the chemokine coreceptor 5, or the CCR5 receptor. So these interactions will anchor the virus to the host cell and stabilize this interaction to allow for the second step, which is going to be fusion of the HIV envelope with the CD4 cell membrane. And this will allow entry of the HIV contents into the host immune cell. Once inside the cell, HIV can use its reverse transcriptase to convert its single-stranded RNA into proviral DNA, which is our third step, which can then enter the host cell nucleus. The next step is integration, where the HIV enzyme integrase will catalyze the integration of the HIV proviral DNA with the host cell DNA. After the HIV DNA is integrated, the host cell's transcriptional and translational machinery is essentially hijacked. So the host cell machinery is now transcribing the HIV RNA that is needed to make new viral particles, as well as translating some of this RNA into long protein chains of essential HIV enzymes. So in order to assemble new HIV particles, the HIV protease must come in and process those long protein chains so that the smaller HIV proteins can be combined with the HIV RNA to form new HIV viral particles. So the newly made HIV virus then pushes or buds out, which is our last step, of that CD4 cell to then go on to infect the next immune cell. So the fate of the CD4 cell can then range anywhere from cell lysis to the induction of apoptosis, leading to a decline in the CD4 positive lymphocyte cellular population, and eventually immunodeficiency and potentially AIDS and potential infections with opportunistic pathogens. Moving to our next slide. So throughout the HIV life cycle, there are a number of viral proteins and enzymes unique to this virus that are essential for this process to occur. This means that we have a number of therapeutically targetable options that will hopefully be specific for the HIV virus and minimize any off-target effects from altering host processes. So keeping this life cycle in mind, let's move on and discuss the 6 different therapeutic classes of antiretroviral agents or ART therapies that are used for the treatment of HIV infection and prevention of downstream consequences. So moving to our next slide. And we will go through these agents in an order that follows this life cycle. So the first group of agents we're going to discuss are going to be our chemokine coreceptor 5 or our CCR5 antagonists. And the example here would be Maraviroc. And this is going to be the only agent we discussed today that was intentionally designed to bind to a host cell target. And as you may have guessed, this agent will bind CCR5 on the host CD4 cells to block the binding of the GP120 subunit of that envelope protein complex. So without this binding, fusion and entry of the virus cannot occur. So because of its unique mechanism of action, it's going to retain activity against viruses that are resistant to other agents. However, this is only going to be effective for CCR5 tropic strains. As for pharmacologic considerations, it is a CYP3A4 substrate, so care must be taken with CYP3A4 inhibitors and inducers. And this agent does come with a black box warning for hepatotoxicity. That is preceded by a severe rash, so counseling to watch for this will be important. The next agent on this slide that we will discuss is a fusion inhibitor. And the example here is enfuvirtide. And this agent inhibits the fusion of the viral and cell membranes by interfering with the GP41 and CD4 interactions. So CCR5 is not involved in this mechanism. But by interfering with this particular interaction, fusion and entry of the HIV virus cannot occur. So this agent will also retain activity against viruses resistant to other classes of ART. Again, because it has this unique mechanism of action, but it is not going to be effective against HIV-2 infections. So this agent is actually a synthetic polypeptide. So it is not going to be able to be administered orally or PO. So it's going to have to be administered sub-Q and usually twice a day. And because of this inconvenience and high cost, and it does come with a risk of cutaneous toxicity, it is generally reserved for those that have failed other agents. So now that we've addressed the entry inhibitors, now let's move through the life cycle and discuss the agents inhibiting HIV reverse transcriptase. So we'll move on to our next slide. Now the next 2 groups of agents we will discuss are targeting HIV reverse transcriptase, and these fall into 2 different groups. The first are going to be the nucleotide reverse transcriptase inhibitors, or the NRTIs, or the second group, which are the non-nucleoside reverse transcriptase inhibitors, or the NNRTIs. And you can probably guess what the difference is going to be. One is going to look like a nucleoside-nucleotide analog, the others are not, but we will still go through each of these groups in more detail. So we will start off with our NRTIs, and these will include a handful of agents like abacavir and zidovudine, and several others. And most of these will either end in this INE ending, except for abacavir and tenofovir, which will have these vir endings. Now, these agents are all nucleoside analogs except for tenofovir, which is a nucleotide monophosphate. And these all must enter the cells to undergo phosphorylation to generate a fully phosphorylated analog that can then act as a sort of decoy substrate for the HIV reverse transcriptase and block its activity. And this is through both competitive inhibition of native nucleotides, but these agents can also be incorporated and lead to the termination of the elongating proviral DNA, because these agents are all lacking the structure that is needed to allow for the addition of the next nucleotides to the growing strand. So these agents work for both HIV 1 and 2, as well as some other viruses. As for resistance, because these agents work at the catalytic site of this essential enzyme, resistance develops much more slowly. But if it does occur, there is a risk of cross-resistance, but usually this is confined to those with similar structures. Like if resistance is going to develop to a thymidine analog, then the cytidine analogs may still work. As for pharmacokinetic considerations, these agents are not really involved in pharmacokinetic drug-drug interactions like some of the other agents we'll discuss in this video, because they are primarily renally excreted and go through glucuronidation. So these agents are not CYP substrates. As for toxicity, this is really going to depend upon how selective these agents are for HIV reverse transcriptase versus host DNA polymerases, specifically polymerase gamma, which is a mitochondrial enzyme, and the one we are most concerned about inhibiting with these agents. So for those agents that are capable of inhibiting polymerase gamma, there is going to be a risk of mitochondrial toxicity. And may lead to anemia, neuropathy, myopathy, and there is a black box warning for lactic acidosis. Which makes sense if we have this mitochondrial toxicity. So these effects can also cause HIV lipodystrophy syndrome, which is a type of metabolic syndrome that can occur in a good number of HIV patients, but is strongly linked with this NRTI class with their mitochondrial off-target effects. Again, this varies based upon agent. And lastly, abacavir specifically comes with a risk for a unique and potentially fatal hypersensitivity syndrome that is linked to the HLA-B specific genotype that we discussed with pharmacogenetics. And this will actually require pharmacogenetic testing prior to starting this particular agent. So nearly all patients that are starting ART will be on at least one of these agents. So it's very important to become familiar with this class and their pharmacologic considerations. So now on to our NNRTIs, which are not going to look like nucleosides and include agents like efavirenz and nevirapine. And these agents will all end in this INE ending and have a VIR in the middle somewhere except for efavirenz. And instead of acting as competitive inhibitors of HIV reverse transcriptase, they actually bind to a hydrophobic pocket at a distant site on the HIV reverse transcriptase to induce a conformational change, significantly reducing the enzyme's activity and leading to non-competitive inhibition. So the site that they bind is only present for HIV-1, so no HIV-2 activity. And because they bind to this distant site and not the catalytic site, they are susceptible to higher levels of resistance. As for pharmacologic considerations, these agents do undergo hepatic metabolism, and some are CYP3A4 inhibitors, and some are inducers. So watching out for drug interactions will be important with this class of agents. So rash is very common upon initiation, but there can be more severe cutaneous manifestations like SJS, for example, even though this is rare. And there is a black box warning for fatal hepatitis with nevirapine specifically. So these agents, like efavirenz, are effective and commonly a part of first-line regimens, but in combination with two other agents because of the high risk for resistance. So now that we've addressed the reverse transcriptase targeted agents, let's move on to our last two groups of agents. The next group of agents we will discuss will be our integrase inhibitors. And one example agent is raltegravir. And these agents all end in this tegravir ending. And as you might have guessed, these agents are going to bind HIV integrase and inhibit integrase strand transfer. And basically this means we're blocking the formation of covalent bonds between host and viral DNA. So these agents are very potent and well tolerated with little clinical toxicity. However, they are metabolized by CYP3A4. So special care must be taken with CYP3A4 inducers and inhibitors. And now this brings us to our last group of agents, being our protease inhibitors. And this group is commonly abbreviated as the PIs, which is going to include agents like saquinavir and ritonavir. And these are all going to end in this NAVIR ending. And these agents are all going to mimic a peptide-like structure to competitively bind and inhibit the HIV proteases, which are distinctly different from our proteases, which is important. So this inhibition prevents the processing of that long polypeptide chain, initially translated so that the viral assembly cannot proceed to mature viral particles. So these are very potent agents, but come with a few concerns to be aware of related to their metabolism by CYP3A4 and their ability to inhibit this enzyme to clinically relevant degrees, which is why it can be found in our CYP inhibitor mnemonic, which remember we introduced in blocks previously, which is G-Pacman, and the protease inhibitors are the P in this mnemonic. So their potential for metabolic drug interactions is most concerning, and ritonavir is by far the most potent inhibitor and can actually increase steady state area under the curves or AUCs by 20 to 30 fold for other agents that are metabolized by CYP3A4. So this specific agent cannot be used with drugs that are metabolized by CYP3A4 that are narrow therapeutic window drugs, like fentanyl and some benzodiazepines, for example. And this is actually a black box warning. So because of ritonavir's potent inhibition of CYP3A4, low dose ritonavir is commonly combined with other protease inhibitors, which remember are metabolized by CYP3A4, to essentially boost their levels so that the primary protease inhibitor's dose and dosing frequency can be reduced. And this is called a boosted regimen. And rates of resistance to these agents fall somewhere between the NNRTIs and the NRTIs if the protease inhibitors are unboosted. But this boost with low-dose ritonavir provides an added barrier to resistance. And then just lastly, as we're talking about these boosters, there is one agent that's called sobisistat, which has no antiretroviral activity, but is combined with other agents to inhibit CYP3A4 and boost ART levels. But this time, like with an integrase inhibitor. So I just wanted to make sure that you are aware of this agent if you see it in any of the ART combinations. So moving to our summary slide. So just to summarize a few general considerations for ART. So it's important for patients to really understand the risks and benefits of ART, to set realistic expectations, and so they understand the importance of adherence, because this will require lifelong therapy. And there is a significant risk for rapid emergence of permanent drug resistance if patients are not counseled appropriately and do not adhere to their regimen. So to help minimize resistance, 3 drug combinations are basically the minimum standard of care, and treatment failure is usually the consequence of non-adherence and will likely require a completely new combination of agents. And for these reasons, this is why it's important to become familiar with these different classes of agents and their important pharmacologic considerations so that you can counsel patients sufficiently to promote adherence and improve patient outcomes.